electrophoresis. You'll hear people raving about this one because it tends to be a favourite. Gel electrophoresis is a crucial technique for separating DNA fragments based on their size and charge. So basically, gel electrophoresis works like a molecular sieve, where molecules are sorted based on how far they can move through a gel under electric field. This gel is made up from a porous gel material like agarose, or it's just essentially, it can be any material, but generally that is the common one. It needs to be porous, however, in order for this to work. So you can think of this gel matrix as a microscopic maze. Molecules will travel through it, but at different speeds depending on their size. You can imagine that a bigger DNA piece will struggle to get through the same pore as a smaller one, and that's the logic that's being used here. DNA samples are mixed with a loading dye to make them visible, and then these samples are loaded into small wells or slot at one end of the gel. Once the gel is set up, an electric current is applied to this gel matrix. Because DNA is negatively charged, when the current is turned on, it pulls the DNA fragments through the gel to the positive end, which is opposite to where they were loaded. So smaller DNA fragments move faster through the gel because they can navigate through the pores more easily, and larger fragments move slower because they get caught more often. This separation creates distinct bands of DNA fragments along the gel. After they run, the gel is stained with a dye that binds to the DNA. And this makes the bands visible under UV light or other detection methods. Now, you might be thinking, why is this so useful? And why is it such a favourite? Well, for one, gel electrophoresis is crucial in forensic scientists for DNA profiling. By comparing the band patterns of DNA samples, like those from crime scenes and suspects, scientists can determine matches or exclusions, helping solve crimes and identify criminals. For example, this example on the slide, you can see that this is the DNA found at a crime scene, and these are the DNA from the suspects. The band for the suspect 2 aligns with the crime scene, which suggests that that's their DNA, and suggests that they're guilty. You'll probably do a couple of examples in class for this, but it's a fun and interesting one, especially with, I know a lot of people that have a keen interest in forensics. It's actually also used in genetics to study polygenic inheritance, where traits are influenced by multiple genes. By separating and analysing DNA fragments associated with specific genes or regions, researchers can understand how traits are inherited across generations. The final one I want to mention, in food science, gel electrophoresis is used to test the purity and quality of products like champagne. It can also detect genetically modified organisms by analysing the DNA present in food samples, which ensures food safety and regulatory compliance. As you can see, there is a whole wide range of applications for this biotechnology, and extends into other things besides just biology, which is what makes it such an interesting one.